are all brains homologous? So most neuroscience students just think I'm talking about human brains. Yeah, well, of course, they're all the same. No, the issue is my brain and the brain of one of my flies, is it the same thing? Did the common ancestor of us and a fly, so of a deuterostome, me, and an ecdysozoan, a fly, did that common ancestor, which was maybe about 700 million years ago, did it have a brain? And if it did have a brain that we would recognize, what elements of it are still of that primitive early structure are still present in both our brains?
Hi everyone, welcome to Reason with Science. I'm your host Jitendra. This is a conversation with Matthew Cobb. He is a professor of zoology at the University of Manchester, where his research focuses on the sense of smell, insect behavior, and the history of science. His latest book, The Idea of the Brain, describes beautifully the past, present, and future of neuroscience. In this conversation, we talk about contribution of philosophy and psychology in understanding brain, modeling of a brain, importance of connectomes, understanding consciousness, and future of neuroscience. Enjoy the conversation, share and subscribe to support the podcast. Thank you for listening. Hi, Matthew. Welcome to the podcast.
Hello, Jitendra. It's good to be here. It's good to see you.
Yeah, so in your book, which is The Idea of the Brain, I read a quote which I want to read. In reality, the history of science is not a progression of brilliant theories and discoveries. It is full of chance events, mistakes, and confusion. So first of all, what a great way to look at it. It's amazing. And the second thing, it was really interesting for me to look at your career. So you were trained as a psychologist.
Oh goodness me! Well,
yeah. Worked as on history of science. So what do you think? What's your chance events that you recall? Mistakes and confusion.
I mean, the chance events are probably too many to mention. I mean, most. I mean, I'm old, right? I'm getting on for retirement, and most scientists of my age, I suspect, think that they have achieved their position by virtue of being clever and having brilliant insights and all the rest of it. And to be an established scientist, you do have to be reasonably smart. But I think we all know that most of what has happened to us is chance, and we've been incredibly lucky. And that's one of the reasons why I say to my students that, who are talking, say, about wanting to do a PhD, I say, fantastic, that's great. Or my daughter, who's just started out on that path, fantastic, great, but don't have as your aim, as your career ambition, to be sitting in the chair that I'm sitting in, and therefore, to be a professor and to be a few years away from retirement, because that is just so unlikely. Not because you're not clever, but because there are just so many random events, and academia is even more prone to chance and to luck than probably most careers. I mean, I don't know, you'd probably be better off saying, right, I want to be a Formula One racing driver or a professional footballer. You've probably got more chance maybe of ending up doing that than you have of ending up being an established professor close to retirement. So what have been the chance events in my career? Well, I think the first one would be, so I spent most of my time studying Drosophila behaviour. And that would be the first chance event. I was an undergraduate in 1976. Can't remember. I was just going into my second year, and there's a UK magazine called New Scientist, which at the 1970s was extremely significant in terms of presenting a lot of the latest academic research to ordinary readers. And I read and they had little kind of summaries of recent research articles, and this one was about half a page, and it dealt with a recent study in the USA that had used Drosophila to study learning. And what they had done was to make a mutant that couldn't learn. Now, as it happened, it turned out that it could learn, it just couldn't remember for very long. Anyway, that's the detail. But I saw that and I thought, my God, I want to do that. That is amazing. They have made a mutant, and because the way they'd made it using a particular chemical, they could be pretty confident that it was a point mutation. So a single letter of DNA had altered in these poor flies, and now they couldn't learn. So for me, this was a double revelation. First, that flies could learn. I thought that was pretty cool. And these are Drosophila, tiny, tiny little insects. But secondly, that you could make a mutant that could not learn, and then in principle, it would be possible to get at the proteins involved, at the pathways involved. I mean, all this was fantasy when I studied this. It must be remembered that in 1976, it was still not proven that DNA was the genetic material in eukaryotes. So it's only known, it's only absolutely nailed and demonstrated in viruses and bacteria. But I mean, a year later in 1977, it was demonstrated in Drosophila. But even so, this was a fantasy, the idea that you might use it for that. But that was something that I immediately bought into. So firstly, I was studying psychology. I was interested in psychology in those days, was much more like neuroscience today, except with a bit of kind of Freud thrown in. Yes, I did have lectures on Freud, but my department in Sheffield, where I was quite extraordinary. There were people studying David Marr's theory of vision in computers. There were people studying artificial intelligence. There were people studying electric fish, and so on. So there's a vast range of people looking at trying to understand behavior. And so my first piece of luck was seeing this article, because I might have just not bought New Scientist that week, wouldn't have seen it. Secondly, an extraordinary piece of luck that I had no idea about until the next week, because I happened to be, although I didn't know it, in one of the only places in the UK that was studying Drosophila behaviour genetics. There's only two at the time. And the only place that was actually looking at it using mutants was Sheffield University, in the Department of Psychology and the Department of Genetics, with my two people who ended up being my PhD supervisors and good friends for many years until they died in the last few years, Kevin Connolly in psychology and Barry Burnett in genetics. And the next week, we actually had a practical class where instead of studying human memory and doing tests of attention or whatever, which is normal stuff we did, we got a load of Drosophila that when you, in a bizarre phenotype, if you gave them, if you etherize them, that knocks them out, you give them ether, these flies would start shaking. Now, the basis of that was barely understood at the time. It turns out that the reason why the flies shake under ether is because they got a mutation in a particular ion channel, which is controlling the neuronal activity. So anyway, the point being that we could do these experiments on Drosophila, and I suddenly discovered that this system, which I thought was amazing, I could actually study it in the department where I was. So that's two astonishing pieces of luck. Without those events, I wouldn't be here now. So there you go. I mean, you could go back further. Why was I at Sheffield in the first place? I was at Sheffield in the first place because I didn't get into Oxford. So if I'd got into Oxford, which would have been great, and I was pretty pissed off that I didn't, in fact, who knows what I would have done, but I would definitely not have studied Drosophila behavior, which is largely what I've done for the last 46 years. So you can start there. And then, of course, anybody's life looking back is full of these sliding doors moments where you realize, well, I did that and that was lucky and that was lucky. I went to London and my first postdoc was on, after I'd done my PhD on Drosophila, my first postdoc was on humans. I got humans drunk for 3 years at the Institute of Psychiatry, looking at the effects of genes and environment on alcohol responses. But whilst I was there, we organized the Behaviour Genetics Association conference, and there was a small session on Drosophila, and I met somebody who said, you must come and work with me in Paris. I've got a lab, we study Drosophila behavior, you're exactly the person I'm looking for. And if I hadn't met Jean-Marc Jalon, who ended up being extremely influential in my career. He's the person who discovered really and demonstrated the existence of pheromones in Drosophila. I wouldn't have ended up working on chemical communication olfaction. So every kind of step. One of the examples that I've used to try and explain this to people, this how unlikely it all seems, and yet also kind of determined, is if you think of your very existence. You only exist because your parents, unless you're the product of IVF, but you only exist because your parents made love. You don't want to think about that, but they did on a particular moment, a particular day, and it was your mother's particular egg that month and one of the bazillions of your father's sperm that joined together to produce you. If we're just talking about the genes and the genetic factors, that is incredibly unlikely. And then all the events that happened when you were in your mother's womb, I don't know, she listened to jazz, she ate curry, or whatever, whatever environment she provided you with, the language you heard as you were a growing fetus, all that altered you. And then you pop out and all the 1,000,001 cultural effects that alter you, form you as being you, contributed to that as well. So what looks from our standpoint is kind of inevitable, we're all inevitable, is in fact just this astonishingly unlikely series of events. And science, being just history, is no different from product of culture, is no different from anything else in that respect. There you go, I've gone on too long, but that's a lot of examples for you.
No, I think, yeah, this definitely makes sense. Actually, a couple of days back, I was having a conversation with Robert Plomin.
Oh yeah.
And this is what we were discussing there, because his work is kind of put in a way that it's a genetically deterministic view, but it's not really. It's the only fact that we don't understand this environment, this kind of luck or chance or whatever we can call it. These randomness or these events that we don't understand in life.
Yeah, they're extremely significant and very difficult to plan in advance because stuff happens. And so my advice to young scientists is do what you enjoy, don't have a massive plan, have many plans, because you may, in particular these days where science has turned into a bit of a huge Ponzi scheme and we churn out PhDs and postdocs and then people like me don't die or retire. That's especially the case in the USA. It's not so much the case in Europe, where obviously people die, but retiring. So in the USA, I was at a meeting the other day and it was full of men in their late 70s, early 80s bemoaning the situation and how terrible it was that, people couldn't set up a lab until they were 45 or 50 in the US. They get their first independent grant when they're 45. That's the average age now. When these old blokes got it, it was 27. But they weren't retiring. They're just carrying on working. So have lots of plans. That's one of my advice, and be prepared to move out of academia because, other life-affirming careers are available.
Yeah. And that's certainly, I think, important. The fact that we can we should think of different alternatives. And that's also kind of a sign that we see in the history of neuroscience that you have discussed in your book. And for me, it was interesting to read this view that for centuries we, like humans, were just focused on this heart-centered view. This is just amazing, and that how much we can learn, and now it's very difficult to think that at some point our ancestors thought that heart is the major center of action.
Well, there's a very easy reason for that. It makes sense. If you think about it, it's not surprising that people thought that the heart was the center of emotions and feelings, because that's what it feels like. You might have the sense of having a little person in your head peering out of your eyes, but all of your emotions, all of the interesting things that happen to you happen in your body. Yeah, your heart starts pounding when you're excited. If you're frightened, your guts start churning. So it was quite logical for people to imagine that some other part of the body, if they thought it was in a part of the body at all, was the location of feeling, emotion, consciousness, and so on. So our interest in the brain is really recent in historical terms. And by looking through an awful lot of anthropology, it was clear that there's no people that I have discovered who have spontaneously, as part of their culture, come up with the idea that your ideas are in your head. And there's quite the opposite. There's vast amounts of evidence that most people in most parts of the world have thought it's the heart or the liver, or most interestingly, and this isn't, I don't think it's mentioned in the book, because only barely in passing, as I was getting towards it was in the final stages of writing, I realized that although I'd looked at lots of evidence from anthropologists, as I say, from around the world, and I was very keen to get away from a European scientific perspective, I didn't have any evidence from, well, there's no evidence from Africa, partly because these cultures were destroyed by Western influence, or their legacy cultures were destroyed very, very quickly. And you can't get back to what people thought before they came into contact with the ideas about the head and so on. But I didn't have any information about Australia. And there we know exactly when we encountered those people. And also there's been a lot of information. And I couldn't find anything at all. I got a big encyclopedia of Aboriginal thought, and brain isn't in there at all. Got it out of the library. It's very excited. Not mentioned at all. And I couldn't work out why. And I emailed an Australian Aboriginal academic, and she said, well, if you asked an Aboriginal person where their mind was, it wouldn't mean anything to them. It's not a question that would have any sense, because their view of themselves is linked not to bits of the body, but to the land. They are part of the land. And this is a key aspect of Aboriginal culture and thinking and views of the cosmos, that people and the land upon which they live and roam are as one. And so you can't just separate, they're not going to locate it in part of the body, because it just doesn't mean anything. So I thought that was very, very interesting. And it shows that these deeply held views we have are very different across the planet, but they make some kind of sense. Because one of the things that I've learned, and I tell all my students, is that in looking at scientific views from the past, you are never, ever allowed to say that people were stupid, right? You cannot allow yourself for an instant to think that past views were stupid. You can think they're silly and funny, and often they are by our standards, but you have to take them utterly seriously, because the people back then are just as smart as we are, and in most cases, and that's why their writings have survived, they were far smarter than most of us, and yet they believe what looked to us like bizarre things. So then the trick, or the challenge, is to work out why. Why did they think that? Why didn't they realize what we understand? And that also, of course, does have a kind of hint of modesty, because you can expect that in a few centuries, people will be looking back with, at the very least, mild amusement, sympathy with our ignorance and our struggles, and possibly be guffawing at the ludicrous things that we think are true.
Yeah, but then, so the scientist Galen, he already kind of showed around that time that it is the brain where all the action is happening.
Well, yeah, what Galen did. So Galen is mainly known in the West these days as the man who helped to synthesize the humors theory of behavior. You have different humors: black bile, yellow bile. I forgot what the others are, and there are 4 of them. And if you don't feel right, it's because your humors are out of balance. But he was so much more than that. He was a poet, he was a philosopher, he was a physician, he did lots of dissections. And he also did this experiment, this rather gruesome experiment, which I'm not going to describe in any detail because it's rather unpleasant. But basically what he was able to do, and this was well known and was described, was to show that if you push on a pig's brain, it goes unconscious and stops making a noise, whereas if you hold its beating heart and stop it from beating, the pig is still conscious and will carry on making a noise. So that clearly demonstrated that at the very least the production of the voice, the noise that the pig was making, was controlled by the brain was fundamental, not the movement of the heart. And that was possible to elaborate that into a general theory that the brain was fundamental because the dissections of occasional dissections of humans, which for a very brief period were allowed in Egyptian, in Greek-controlled Egypt around 200-300 years before the Common Era, they showed what's fairly kind of obvious, that all your sense organs go to the brain. They don't go to the heart. There's no those nerves, those neurons, as the Greeks called it, which just means sinew. Those neurons were connecting the eye to the bits of the brain. They didn't go down to the heart. So there was kind of good anatomical evidence to suggest that maybe the brain was what was going on, even though it doesn't feel that way. And what Galen did was this rather neat experiment, if unpleasant, and that was known. That was well known, well into the, I mean, throughout the in the West, and then as Greek ideas were preserved and developed by the Arab world, and then the West rediscovers them in the very beginnings of the Renaissance in kind of the 12th, 13th centuries. These ideas are known, but because of the weight of other ideas from Aristotle, for example, who argued very clearly that it's the heart for all the reasons I've explained, and that the brain was just some kind of cooling mechanism, people thought, well, Aristotle is pretty smart, and everything else he says is basically right, so we'll go with Aristotle, because, you know, okay, Galen's experiment, who cares, one experiment, you know, the experimental proof didn't have the same weight, epistemological weight, that we would now give to it. So it wasn't like, okay, here's the killer argument. And indeed, there was no brain-centric experiment. There wasn't a single thing that made people go, okay, it's the brain. In fact, it's just a slow accumulation of evidence, more dissections, more elaborate dissections in the 15th and 16th centuries. It becomes really, really apparent that the brain is unbelievably complicated, whereas the heart, as William Harvey put it at the beginning of the 17th century, the heart's a pump, or as Steno, another anatomist, put it in the middle of the 17th century, it's a muscle. That's all it is. You know, it doesn't do anything else. The brain, on the other hand, is mind-bogglingly complicated. So towards the end of the 17th, beginning of the 18th century, everybody in the West who's educated comes to accept that it's the brain that's doing the stuff. But to close on this, what's fascinating, you can see this uncertainty in Shakespeare. In Twelfth Night, in one of his the little songs that intersperse his plays, somebody sings, "Tell me where is fancy," that is imagination. "Tell me where is fancy bred, where is it born? Or in the heart or in the head?" So what's striking here is not only that Shakespeare knew that all these clever people weren't sure, and that it could be in the head, and that nobody knew, but he also knew that he could make a kind of not particularly funny joke about it, and the groundlings, who are the people who would be standing up and throwing cabbages at the play if they didn't like it, those people, the ordinary people who went to see his plays, they would understand it too. So this isn't just an abstruse debate amongst, you know, the elite. It's also something that people are interested in for fairly obvious reasons. It's kind of fundamental question of existence. What is it what is it that makes me me?
And I think that also, that at this point, I think we can kind of start thinking of philosophy in general, like what was the contribution of philosophy and what is the contribution of philosophy in kind of making neuroscience more enigmatic, or, you know, like.
Yeah, I mean, Francis Crick had a very neat phrase, which I have adopted. You should listen to the philosophers' questions, not to their answers. And he spent a lot of time hanging around with philosophers in the last kind of 20 or 30 years of his life. I'm not really sure, you know, that philosophy did play a massive role in helping us understand what the brain does and how it does it, because although it was very significant in keeping in the 17th and 18th centuries, keeping the question of consciousness, of how it works, at the center of what we would now call scientific thought. So thought based on ideas based on experimentation and so on. I'm not sure that really the philosophers contributed a massive amount. The main way, for example, that Descartes contributed to thinking about the brain was in terms of his hydraulic theory of how it worked, which wasn't a philosophical theory at all. It was a scientific theory. It was based on observation. He noticed these statues in Parisian parks that moved in a kind of crude animatronics, and they all work by weights and by water power. And he thought, well, maybe that's how the body moves. That's how the brain works. There's kind of this power of hydraulic stuff going down our nerves. When this was eventually published after his death, it's published in 1660, it was immediately demonstrated that he was wrong, because all you had to do was, get any old bit of animal flesh after it had just been killed and then chop a nerve, and nothing came spurting out. There is no hydraulic fluid inside a nerve. So people fairly soon went, well, that was a nice idea, but it's wrong. And what's striking, it is wrong, but it was productive, I think, in that it is the first real example we've got of people trying to use metaphors about technology to explain how the brain works. But I don't think that didn't help. That didn't come from Descartes' musings about the nature of spirit and mind, and how you could have an immaterial matter, which is what he thought that the thought was, and the qualitative differences that he argued between animals and humans, because he located this in the pineal gland and said, well, clearly the location of the soul, or the point at which this immaterial stuff that is the soul interfaces with the brain must be the pineal gland, because only humans have the pineal gland. Well, again, it only took a bit of rummaging around in a few dead animal brains to show that lots of animals have pineal glands. So I'm not really convinced about the role of philosophy, I'm afraid. It had some, yeah, there you go. I think I'll just leave it at that. You have to talk to a philosopher. There are plenty of philosophers who get very cross about that. But one of the reasons for that is that it's going to sound kind of shocking, but the main thing that philosophers are interested in terms of the brain and so on, is consciousness. And I'm not very interested in that. Not because I don't think it's amazing, but precisely because it is so amazing, I don't think it's a doable problem at the moment. And I've thought this since I was an undergraduate, because people get very excited about this now. And that's all the fault of Francis Crick, who kind of made it a scientific issue. Because it wasn't, when I was an undergraduate, it wasn't something that anybody was interested in, because it was not, we couldn't see how to study it scientifically. And that is still the case. I think Crick's approach was very powerful, but there are a million and one theories of consciousness. And I don't think I don't think any of them are anything more than, they're like the old joke about the man who's lost his keys at night, and they're groping around he's groping around the base of a street lamp, and somebody says, "Where, you've lost your keys?" And he said, yeah, I can't find them. Why are you looking there? Did you drop them there? No, I dropped them over there. Well, why are you looking there? Because that's where the light is. And I think that's basically where we are. They've got some idea and they're rummaging around, but really, the real answer is way over there, or nowhere, or distributed.
Yeah, that's actually shocking. That you are not interested in consciousness, and we'll get back.
Well, I'm not interested. It's that I'm a scientist, so I'm not. A philosopher, it's absolutely fine, and philosophically minded neuroscientists. I have no problem with people doing it. It's just that, I don't think we've got very far. And I think we've lost sight. Crick's insight, it's all Crick's fault. If he hadn't decided that he was going to look for the neural correlates of consciousness, it's very significant. That doesn't mean to say consciousness. He wasn't trying to come up with a theory of consciousness. Quite the opposite. He didn't think that was going to happen for a century. And Crick's whole method was to ignore stuff that would get in the way. All right. So when this is what led him terribly astray in the early days of trying to find the structure of the DNA. And he and Watson came up with this absurd three-strand helix theory, because they were just trying to ignore a load of stuff. That, well, let's deal with the simple stuff. But that's the method that he always had. And he uses that to try and understand, or to begin a research program on the brain as well, which turned out to be incredibly influential. But I think we've kind of lost our way. in two respects. One, that people aren't looking for those correlates, they're looking for the thing itself. They're trying to come up with a big complicated theory that can explain stuff, which just can't, any of the myriad theories there are. Secondly, the other thing that characterized Crick's ideas from the very beginning is reductionism, is trying to find explanations in simple systems, which you hope will eventually provide an explanation in more complex systems. Now, you know, one of the things that the theme of the book is that we don't know how the brain works. It's not just that we don't know how the brain works. We don't know how the simplest of nervous systems work. We don't even, it's not even, we just don't know about the nervous systems. We don't know. So the example I give is Eve Marder's work. Eve Marder is at Brandeis University in the US, and she has spent her whole career studying the lobster's stomach. And the lobster's stomach has just 30 neurons in it, and this these 30 neurons produce two rhythms pattern rhythms of activity that activate the muscles that grind up the lobster's food. So those 30 neurons, she knows everything about them. She knows their connectomes, she knows the genes that are expressed in them, she knows everything. And yet she can't explain why those 30 neurons produce those two rhythms rather than any other rhythm, which the models say they could do, nor why you know, and some other set of neurons hasn't evolved to produce those two rhythms. So for me, that is the challenge. And as clearly as somebody who's studied Drosophila for over 40 years, I think that explaining things in simple systems is the way to go. Not necessarily Drosophila, it could be in a larval zebrafish or whatever, but something with only 10, 100,000 neurons, perhaps, but 80 billion, doing the most bizarre, complicated thing in the universe, thinking. I think that's the wrong place to start. There you go. We got to the end at the beginning. Sorry about that, but I thought it best to explain myself very clearly why I'm not interested in it. I just don't think that it is a doable problem. I'm quite happy for people to study it. I'm not sure if I was on a grant proposal committee and somebody said, I want to work on it, I'd give them any money, because I'd give the money to the lobster's stomach, not to the theory of consciousness.
Yeah, so so as you mentioned about the reductionist approach, I mean, that's one way to look at it. I think the other thing that we need another reductionist approach in the neuroscience today. You know, first of all, like to start kind of removing the concepts or the ideas that they don't really make sense, you know, and also to understand which part of the science that really kind of contribute in the progress. I mean, the fact that we can kind of say that philosophy hasn't contributed much means this should give an idea to philosophers where they can focus more or how they can really contribute more. Well, and the other,
I mean, I think philosophers can contribute, but it's like explaining what is good to us, you know, what is happiness, that they're superbly qualified to do, and I'm not. But you know, explaining how the brain works, I think, is probably not really their territory.
Yeah, I mean, I think only one point where I generally think where philosophy can make a contribution is the fact, like, if they start looking at the from the science perspective, basically philosophy of science, and they start looking at this metadata analysis, and because this is what we are also doing nowadays, generating this huge data, and of course we'll need, I think, another field of scientists just looking at that metadata and, you know, kind of trying to make some understanding out of it. And this is where I think also kind of philosophy can contribute and help us formulate good theories which may help us to make sense of the most complex thing in the universe. So but before that, what I wanted to say is the other thing that we should highlight here is the fact that, of course, we do science, and as human species, we have a language. And this is where we kind of end up adding a lot of jargon to the work, and especially where we don't understand, right? So this is something also that we can kind of expose the jargon.
So what give us an example. Give the viewers
For example, in psychology, if we see that now there are like different branches of psychology itself, right? We can take the example of Freudian work or like Freudian view of science, psychoanalysis. That's one example. I don't know how much this contributes, especially when we look at from the evolutionary point of view. So what's your view on on these topics?
Well, Freud. I mean, as I say, I've read Freud. I've wrote an undergraduate dissertation on penis envy. So I know all about that. And whilst it's been incredibly influential in the 20th century, in particular in terms of literature, in terms of understanding, providing a general framework, which of course Freud didn't develop on his own and wasn't the only person, in terms of unconscious motivation. So we may do things for reasons we're not aware of, and some of those reasons may be deeply rooted in past experience and trauma or thwarted desires. I think to go from the rest of his framework is bunk, and all the empirical evidence shows, well, it's just nonsense. I mean, his whole. I'm not going to, let's just leave it at that. There is no evidence for it. And indeed, it's very striking that, we're all talking about mental health these days, and virtually nobody says, yes, what we need is more Freudian psychotherapy. Well, no, what we say is we need more talking therapy. We need things like CBT, which are experimentally demonstrated. So I think, Freud was extremely influential in that respect, in terms of making us think about these, and as I say, literature in particular and art, amazingly influenced by these ideas, and that insight that we may do things for reasons that we're not aware of, but beyond that, I'm not sure he made any particular contribution to our understanding of anything. And his work on the brain was, which I occasionally have in the past have got into arguments on Twitter with people about, maniac Freudians. Nobody's writings on the brain were absolutely marvelous. They really weren't. He wrote one very strange document in a kind of fever dream that he later kind of said was not right, and it certainly wasn't. But his whole, though he's trained as a physician, obviously, I think his contribution to brain science is pretty minimal. I think there are, however, concepts and ideas that we can usefully think about, but they're far more basic than that kind of thing. And they're so the idea of representation. Does a sensory neuron, is the activity of the sensory neuron representing the outside world? Is there a correspondence, a direct one-to-one correspondence, or not necessarily one-to-one, but a consistent, is there a consistent correlation in Crick's terms between the activity in the outside world and the activity of that neuron? And whilst on one level, well, yes, because that's why actually you get a load of spikes firing in the neuron when not connected to sensory receptors that detect light, you turn the light on, you get a load of spikes. But in what way is it useful to think of that as this simple one-to-one representation? And literally an impression, you've got to remember the word impression, where that comes from. It comes from the idea that there's something in the outside, comes from the 17th, 18th century. There's this force that is pushing on our nerves, making an impression. So you've got the thing out there, and it alters the shape in some way of the nerves, and then ultimately the brain corresponding. So it's literally, it's an impression. And there are those who suggest that even ideas like that, fundamental ways of thinking about sensory neurons, we need to think more carefully about the words we use. And I've got time for that. And those are from kind of philosophically minded neuroscientists. And I think that will be useful in making us think about what the spikes we're looking at. And that's one of the things that I do, is to look at sensory neurons. What exactly does that mean? Why is it the shape it is? What is the correspondence, the relationship between the stimulus or the thing in the outside world, let's put it that way, that has caused the activity in the neuron, and that activity itself? And then you can start to think about, well, what then happens in the brain when that arrives in the brain? How does the brain then interpret that, if that's the right way to think about it?
Yeah, the other point would be to talk about metaphors, because
Yeah.
science does work in metaphors, and neuroscience is populated by metaphors. So, what is the role of good metaphors, and or are there some good metaphors? What do you think?
Well, yeah, all metaphor. So, what is a metaphor analogy? So, I mean, this is something that philosophers have indeed got very excited about and spent a great deal of time in the last, in particular, last 30, 40 years, thinking very hard about and looking at different scientific traditions and trying to see how they rely upon metaphor. And the obvious way that has happened in neuroscience or in understanding the brain, this is the kind of the theme of the book. It didn't start out that way, it must be said. So my starting point was not to write a book that would be framed by metaphor. My starting point was, to be honest, a bit boring, and it was pretty dull when I was about halfway through it. It was just one damn thing after another, as Henry Ford said about history. And then I realized that by looking at the different metaphors that were being used at different periods, that actually gave an interpretive framework that made it, one, a lot less boring, and actually really quite interesting and exciting even about the future. So if we can just briefly characterize that. You've got Descartes, who looks at the height of technology at his time, i.e. mechanical objects, machines that are working by clockwork or by weights or by hydraulics, and he says, that's how the brain works. And then you get the discovery of electricity in the middle end of the 18th century, and then people start to say, well, the brain obviously works in some way by electricity, and with the mastery of electricity on a continental scale, the development of the telegraph system, then people say, yeah, well, in fact, messages are telegraphed down from the brain to the periphery, exactly like in a telegraph going from point to point. And then people go, yeah, but wait a minute, this is just fixed. There's no flexibility here. In a telephone exchange, this amazing newfangled development, then messages can be rerouted. And that kind of emerges in about 1880. About a decade after the first telephone exchanges. And that, interestingly enough, that is still lingering. I've seen it used in Nature Neuroscience. People describe the brain as a switchboard. So that's clearly a metaphor that has some power, otherwise it wouldn't be hanging around 140 years on. But clearly the brain is not a switchboard in any way, shape or form. And clearly the most recent metaphor we use, and this is kind of the theme of the second half of the book, is the computer metaphor, and the brain is a computer. Now, there are two things to say about that. Firstly, fascinatingly enough, when it began, the brain wasn't a computer, the computer was a brain. So when John von Neumann develops his theory of a digital computer, which he does in about 1944, and that's the computers we're all using now. Every computer, the one in your pocket, the one we're talking through now, the one that people are listening on, they all use von Neumann architecture. And what von Neumann does is to take the paper, very famous paper by McCulloch and Pitts in 1943, which looked at what they thought was the structure of the nervous system, the way that synapses are organized, and says, look, these synapses are basically have a logical structure to them. You could have a neuron that will fire if both two signals come in. So that effectively that neuron is a both, or you could have an or, either one or the other, or a not. So those basic parts of symbolic logic, which we all know about now, and we know are the basis of the computer, was thought to reside in our brains. Now that's complete nonsense, not at all true. But it was inspirational to von Neumann, who said, my God, I'm going to build a computer like a brain. And that was the pitch he made to the US government. He said, yeah, I'll build you a computer. It will be just like a brain. Now, he very quickly realized, because he was chatting to loads of neuroscientists at the same time, that in fact, the brain didn't work that way and it was going to be a dead end. But he did come up with a great idea for a computer that has changed our world. So firstly, interesting kind of quirk of history. It was the other way around. The computer was a brain. Now we've forgotten that, and all we think of is, well, the brain is clearly doing calculations. It's making calculations in the same kind of way as a computer does. So I put out a challenge, not a challenge, a request for information on Twitter. I said, well, can somebody give me a worked example where we know down to the neuronal level? I'm not talking about general, oh, we're doing computations, but actually, an example of an and or then circuit that we actually know and we can nail, and we can actually prove that there's a computation going on. And I think there's kind of 2 or maybe 3 examples in every organism that we know, not talking about the human brain, but in any organism where we can really understand at that computational level. So if you think about it in its detail, the idea of the brain carrying out computations is pretty kind of vague. It hasn't proved heuristic in terms of us being able to work out what is happening at a neuronal level. Now, many neuroscientists wouldn't particularly care about that because they're not interested at that base level. But as somebody who works on Drosophila, that's all I've got. Well, it's a bit more than that. It's not C. elegans after all. It's not just neurons, but it's got a brain. And I need to know at the neuronal level, at the circuit level, how things work and how behaviors are produced. And that's where it gets hard. And that's where we fail, and it's very difficult to show computer-like activity in a nervous system. In fact, everything we know about the nervous systems suggests they're not at all like computers, which have separate modules. And the arguments over localization of function in the brain go back to the 1840s and before. And we're still arguing about it now. And every time people say, yes, it's definitely localized here, and this function, we find it here, it turns out, well, actually, it's a bit distributed, or there are neurons connecting this particular function area with another area which modulate that activity, and it all gets distributed again. So it's very uncomputer-like. So that's the final issue, is, are we coming to the end of the usefulness of the computer metaphor? Has the framework, as I put it kind of glibly, has the framework become a cage? Is it stopping us because we're looking for computations that may not be there? Is it stopping us from understanding something else? Now, I don't have any answers to that, but it's a question that I'm not the only person to ask it.
Yeah, but I think there are 2 reasons why this is happening. First can be simply the confusion between computation and life in general, or biology. So if we think in terms of computation, where of course there is information, and that information
that's a metaphor.
Yes, again.
It's not information in the way that Shannon and Wiener formulated it. That's not in neurons. Neurons don't behave like that. So I'm sorry to interrupt you. It's information in the woolly sense that we use it. Yeah.
Exactly. What I was saying is so there is this digital information, means there is always either it's 0, like no signal, or signal, right? Yeah. That's the computational part. But in biology, the things are not 0 or 1, at least at the molecular level. There are gradients, and
well, it's not, neurons aren't digital. This is what von Neumann realized as soon as he started talking to the neuroscientists rather than talking to the theoreticians, was that neurons don't, they got a strong analog component to them. I mean, that's, exactly going back to representation, what I talked to earlier on. If you stimulate a neuron, a sensory neuron, with differing degrees of stimulus, then you get an increasing frequency of response, at the very least, if not other forms of change. So clearly that's not digital. Each spike is digital. It either happens or it doesn't, but spikes are happening all the time. It's the brain's got to interpret the pattern. That's the key thing.
Yeah, and as you mentioned, it's the distribution, which is what people should think. And most of the times it's true in most of the biological processes.
Yeah, absolutely. Yeah.
And I think the second thing is that our understanding of biology, even, or in science, I think it drives from the engineering perspective that, as Richard Feynman has said, that if I understand well, I can make it. So I think that's also one reason, right? Because we think that probably if we understand brain, we can kind of make it computer-like, or it'll be a computer-like thing, and we can sort of start making these kind of artificial brains and stuff, right?
Yeah, I think that's right. And there's I think that's profoundly mistaken. I mean, AI, again, is not my field, although, as I said, I did study it quite intensively when I was a student, but that was a long time ago. But it's clear that despite the amazing abilities of some of these deep learning programs that we see, and their ability to recognize patterns that we can't detect in massive data sets, and you can, I mean, the most astonishing thing is AlphaFold. So the big problem in biochemistry is how do proteins go from a one-dimensional ACTGCT sequence to a three-dimensional structure in a consistent way, and nobody had any idea, and basically they just chucked all the amino acid sequence and the known 3D structures into one of these deep learning programs, and it came up with an answer. Or did it? Rather, if you now put a sequence in, it will give you a prediction. If you are able to ask it, how did you do that? What rules are you using? The AI couldn't tell you, not because it's an AI, but because nobody knows, including the people who build the damn things. They don't know how they work. This is the most kind of terrifying thing. I mean, not terrifying in the sense that I think they're going to become conscious, but rather that illustrates the gap we have in that these AIs can do astonishing things, but we don't know how they work. We don't know what they're doing. We can't sense what rules they are extracting from the massive data that they're observing, and just because for them it's just patterns of zeros and ones, literally that's all they can see, but they can see patterns that we're unable to. And so it's incredibly powerful heuristic. It can do things and open new knowledge, and clearly for people in protein chemistry this is just unbelievable. It's a dream come true. But if you're thinking we can use that to understand what's happening in the brain, I think it's a non-starter. It's just not working in the same way, and it doesn't actually tell us anything about how to do this thing that brains do. It's doing something else. It's amazing, but it's not a brain.
In the similar lines, we can also talk about this notion that we have over centuries, brain in a vat.
Yeah.
Which is also kind of similar in a way, and that's what also neuroscientists that they're trying to do to for example model the brain and stuff.
Yeah, I mean, I think that, so we're coming back to the starting point, the fact that I think it was in 1997 there was an article in Trends in Neuroscience which had the astonishing title, "The Brain Has a Body," which was clearly meant as a kind of jokey, shocky title. But I think for many people it is neuroscience is a bit of a shock that, well, wait a minute, if it's all going on up here, how come I feel it down here? I feel frightened in my gut. What's going on? I mean, that's weird, isn't it? And yes, it is weird, but it's telling us that we can't. The brain in a vat is a fantasy. And again, it's something that many philosophers, to give them their due, would rail against and say, no, this is absurd, because we are part of that physicality. We are not machines. We are evolved, highly adapted organisms in which, all our neurons are at one end for reasons that are still a bit obscure, actually, in evolutionary terms. When, in fact, what happened in evolutionary terms is when animals developed bilateral symmetry, so you get a left and a right, you also develop asymmetry in your two other planes. So you start to have your mouth down at the bottom because you're a worm of some kind, and you start to have your sensor, you've got dorsal ventral asymmetry, you're not symmetrical in that way, and you've got head tail asymmetry in that you tend to stick your sensory organs at the front end near where your mouth is. And that way you've got bilateral symmetry. You can see which detect a predator or prey from one side or the other. And you've got all the neurons controlling your movements together so you can move more quickly. So, but that shows you that the very origin of the brain, which is still disputed, but the origin lies in evolutionary pressures of the whole body, of the whole animal. It's not, we are not the point of it all. We are just the most bizarre, most recent odd twig on this massive tree, which is all where the brain has evolved to do very, very different things. But it's always about organizing, controlling, if you like, and don't particularly like that, but organizing, coordinating the body in order to be able to do things, representing, I'm happy with that myself, representing the outside world in some way, and predicting, and this is the computational bit, predicting what will happen if the body were to do certain things, if the animal acts in certain ways, what might happen. And that I think is what a brain does. It's kind of, I have a summary slide in the middle of my talk about this. We say, well, okay, this is where we think, this is now where we've got to, which is where we've been for kind of the last 70 years. So it's all happened, interesting, it all happened in the middle of the 1940s. This is when, Kenneth Craik, who was a Cambridge psychologist, he comes up with the idea of the brain having a model, effectively, of the outside world, representing it, the outside world in some way, and being able to manipulate it. And at the same time, you've got theories about information, so the word really becomes very popular. And you've got McCulloch and Pitts with their rather bonkers, but very striking and interesting idea about the brain having a kind of, the very basis of it having a what they called an immanent logic. So the way our brain and nervous systems are wired up, they claimed, it's not true, is based on this if, then, or not kind of logical operators.
There is another kind of now, I hope that more neuroscientists, they are adopting this view, which is neuroscience needs evolution. So I had one conversation with Paul Chisek. He's a professor in University of Montreal. And this was the title of our conversation, because he is looking at the evolution of the brain, the neurosystem. And then also, like, he has his favorite species are like lamprey and these kind of
oh, they're far
like,
too complicated. You got to go further back than that. I mean, they're all vertebrates, you know, they're chordates anyway. Yeah, I mean, that's absolutely true. And so what's one of the fascinating things that the public doesn't realize, and I guess a lot of neuroscientists don't, is, you know, and I've bemused generations of students by giving this as an essay title, and I've learned not to do it because they never understand. Are all brains homologous? So most neuroscience students just think I'm talking about human brains. Yeah, well, of course, they're all the same. No, the issue is my brain and the brain of one of my flies, is it the same thing? Did the common ancestor of us and a fly, so of a deuterostome, me, and an ecdysozoan, a fly, did that common ancestor, which was maybe about 700 million years ago, did it have a brain? And if it did have a brain that we would recognize, what elements of it are still of that primitive early structure are still present in both our brains? Because part of the problem with this is separating out what are conserved features or consistently emerging features, which are rooted in the original, well, ultimately the original genes that are present and the way that development takes place, and what is contingent on convergent evolution. And so the key example of this is in my field, which is olfaction. So when I explain to the public, why do I use Drosophila larvae? So that's the maggot, you know, it's a tiny little thing. You've got 4 million neurons. A maggot's got just, smell neurons, that is. A maggot's got just 21. But the wiring diagram, the first stage processing that takes place in the brain, in a human and in a maggot, of the olfactory signals is pretty much the same. Now, that was long thought, and we used it as an example of conserved character. It's almost certainly not. And if you think about it, well, yeah, of course it's not, because that wormy thing that is our common ancestor from 700 million years ago, it didn't have an olfactory processing region. It wasn't, whatever brain it had, it wasn't as complicated as that. So as putting the brain into evolution and thinking about its deep roots enables us to begin to tease apart, I don't think we can solve, but we can begin to tease apart some of these issues about conservation of characters and convergent evolution. In the case of the olfaction, it's probably the case that that wiring diagram is simply the best way of processing complicated chemical signals. Maybe there's something in the genes that connect, that drive the neurons and make them connect in certain ways that leads to that as well. So it might be something, you know, emergent out of the developmental process. But I think you're absolutely right, yeah. So the brain needs to be put back into the body. We need to understand, and into evolution is that great phrase of Theodosius Dobzhansky, that nothing in biology makes sense except in the light of evolution. And that applies above all to the brain, because so many people, if you say, you know, you're right, I mean, you know, my book is the idea of the brain. It's not about human brains, but it is largely, because that's mainly what people have studied. But I'm not, you know, I've grown out of being interested in humans. They're too difficult to work with. You can't kill them when the experiment goes wrong. Whereas flies, you have complete liberty. So the, you know, all brains, I think, are amazing, because they enable the animals to do all these astonishing things, from learning to courting to moving to eating, you know, everything that they do. The human brain is just the most extreme and mind-boggling version of that. But for reasons I explained earlier, it's not my preferred focus of interest as a researcher, that is.
One of the other interesting aspect is this fact that we have different brain areas for different functions, you know, so kind of this topology
ish. I mean, yeah, it's, you know, it's hard, isn't it, though, when you actually, I mean, it is at one level. So I mean, I was very surprised to read this, because I didn't know. I was rereading Hubel and Wiesel's first papers. So Hubel and Wiesel have got an anesthetized cat. Its eyes are propped open. They've got electrodes in its visual cortex, and they are trying to find stimuli that will activate parts of the cortex. And they do indeed eventually find it. And they're able to show that as you go down through the cortex of the cat, that there are areas of the brain that will detect different slopes of line. And this gave rise to all sorts of rather superficial theories about how we see, that we can see by constructing lines put together and, oh, you've got an image. Now, it doesn't work that way, and it's fairly soon realized. But what's fascinating, that they're in the visual cortex, they're recording from these neurons that detect visual signals, very precise dots or lines. And then they find in the visual cortex cells that respond to sound. So the visual cortex is not only responding to vision, and the auditory cortex is not only the only place that you can detect sound. Sound is also processed in the visual cortex. And just remember, this is a cat. What do cats really, really like? They like sound and movement. You know, they want a rustle in the leaves and a movement, and then it stops, and then they can jump because there's a mouse in there. So what's astonishing is that from the very earliest stages of peripheral processing of things that we think we understand, like visual system or the olfactory system, we now know there are top-down modulations of that immediate peripheral activity by other sensory systems, which is reflecting the evolutionary past of the animal that you're investigating. So our sense of smell, you can, I've forgotten the name of it. There's a substance which you smell it, and you can't quite identify. If you are given a picture of milk, then you go, it smells of milk. If you're given a picture of ham, they say, it smells of ham. And this isn't an identification thing. This is not, you know, some higher, I associate that with smell. It's what you think it is. It's your immediate perception. It's going on in your olfactory bulb. And yet that ambiguity, it's a classic illusion, you know, figure ground kind of ambiguity thing, but it's in the olfactory mode. We now know there are these neurons coming down from the visual cortex and elsewhere, which are modulating our immediate sensory experience. So yes, the brain has localized bits doing things, but that's not all they're doing. And other things are going on in there as well. It's not like your computer, you know, your computer, your hard drive is, or your SSCIs, or whatever they're called, they just do that. They just hold your memory. Yeah, your whatever you've got for your screen is your graphics card is just doing that. It's not going to suddenly start processing sound. They are all absolutely discrete. The brain is not like that. It is only like that at a first degree of estimation. And it is the final example I give on this. I think it's quite amazing. So the strong argument for localization came very early on, as people were developing. As I say, this argument took off in the 1840s, in particular in France, because Descartes had said that thought was unitary, the mind was one, and that therefore people argued, as they're investigating the brain, well, the brain must be uniform as well. And if you look at it, it's the same on both sides, for example. You look at it with a microscope, you can't see any difference in the structure on either side. And then to his great embarrassment, French scientist called Broca discovers that a man who was in his clinic who couldn't speak properly, all he could say was tan. He died of other diseases, and Broca dissects, does an autopsy on him, and he notices that there are substantial lesions in the front left part of his brain. So he notes this. And then there are other patients who have similar, they've had strokes, they've lost the power of speech, who can't speak properly. And he notices consistently it's in the front left part of the brain. Now, he didn't like that because he's a good French scientist, and Descartes had told him that, or thought the brain is uniform. It must be, because thought is. But the data drove him to say, well, actually, folks, there is some localization of function and the front left side of the brain. And we are very, very confident of that. That is absolutely real. You get a magnet, big electromagnet, stick it on the left hand side of your brain when you're speaking, and you can't speak anymore. There are loads of fantastic YouTube videos of this happening. So the one bit I think everybody agrees on is the localization of function to Broca's area. And we don't know how that works and why it works. In the split brain subjects who've had their corpus callosum separated, the right side of the brain can't control speech because it doesn't have access to Broca's area. So the two sides of the brain, one can speak and the other can't. And yet, so that we're confident that's absolutely nailed. There's a big difference between humans and monkeys and all the rest of it. And yet there are people wandering around who have no left side of their brain at all and yet can speak. So there are two cases that I know. One was a young girl in the UK who had cancer and had the left-hand side of her brain excised when she was about 3. She's now at university, can speak perfectly normally. The other was one I saw, a paper just came out, I saw it on Twitter about 2 weeks ago, of a woman who's in her 50s now in America. And when she was in her 30s, she had a routine brain scan. And they said, has anybody ever asked you about your brain? Because literally, she has no left side of her brain. They think she must have had a stroke perhaps when she was still a fetus. So there's nothing there. And yet, because these things happened very, very early on in development, when they were still children or before they were even born, in ways we don't understand, the brain has organized itself to be able to control speech, because that's one of the main things that people do. So there's a kind of, whether it's from the external pressure or from some emergent developmental factor, the brain's found a way of being able to make speech in a way that is completely undetectably different from the way that you and I do it. So even that fundamental localization is not always true. So, at one point I did think about writing the whole book about localization of function. I thought, no, that's a completely different book, and I have really no idea what I think about it, because we want to localize things. That's what the reductionist tradition does. This is happening here, and then we know about it, but it's not as simple as that.
Well, one reason is the fact that, brain, it looks like this rigid blob kind of thing, but at the end, there is a lot of plasticity, and this we also need to understand and appreciate, right?
Yeah.
And this plasticity comes from, what do you think, from cellular interactions or?
Well, development, to start with development, identical twins don't have identical brains. They don't have identical bodies in all senses. They're not identical. They've got same genes, but because stuff happens, and we're not talking about, oh, well, they were separated at birth or anything. Just two people who live exactly the same lives don't live exactly the same lives. There are stochastic processes in development that mean that different lineages will dominate. But this is the case even in, C. elegans, which Well, C. elegans people get cross when I say this, but it doesn't really have a brain. It's got about 8 neurons that are connected together. You know, it's got whatever it is, 302 neurons in total to do everything. But it really doesn't have much of a brain. And yet, C. elegans are not identical. They're not robots, you know. You drink a can of Coke, it's always the same. Robots do the same thing. Organisms are alive and have got a developmental history as well as an evolutionary past. And even being reared with identical genomes, reared in identical environments, like you can do with animals, experimental animals like flies or worms, they're not all absolutely identical. So I think there's a lot of noise in the system, and that's partly what produces individuality, personality, if you want. I'm quite okay to imagine, I don't think it's been demonstrated really that worms have personality, but I imagine they do. And personality is just a consistent way of behaving. The simplest way of imagining a personality in animals is what's called bold or shy. So an animal that is bold, if you put it in a particular circumstance, an arena or on a plate, will move more quickly than an animal that is shy. Now, I don't think that's got anything to do with being bold or shy in humans, and you can understand it simply as a latency. So how long is the animal going to sit there before it decides to move? And that latency can be relatively consistent in a same organism. And that's the basis of personality. And that, I suspect, will emerge from stochastic developmental processes. A bit like handedness. You know, handedness, there's no gene for handedness. It's constitutional. It's absolutely real. It's hard to imagine being the other-handed. And yet, where does it come from? We still don't know.
What's interesting here is the fact that, so, I mean, of course, all of us, we have this 98 to 99% of the genome, which is like the same in all humans, right? And which gives us a certain characteristics, which makes us human. I mean, like most of the people, we have both the hands and the faces like in the same way, and the eyes are there, and the similar kind of thing. We can talk about the anatomies of our different organs, right? And then these stochastic processes are working on top, which basically makes us different in a way. And when I think of that in the brain's point of view, it's the connectomics part is interesting, right? Like how do so one thing is that we get the connectomics, but what is the basis? What we are looking there, you know?
Yeah. So I mean, connectomics, and again, this is all down to Francis Crick. I mean, so when I was writing before I started writing the book, my impression as an undergraduate, because I was an undergraduate when Crick left the UK to go to California, partly for tax reasons and because he was going to be made to retire when he was 65 in the UK, and he didn't want to do that. He wanted to carry on researching. So he went to California. And, you know, the joke, not the joke, but what we said is, yeah, so Crick, clever guy, goes to California, tries to solve consciousness, ha, ha. And that was my impression when I went into writing the book. However, it turned out that in all the book's divided into, well, 3 halves. So there's the past, the present, and then a tiny third half, which is the future. And the present part is composed of, I can't remember, 7 chapters, something like that, dealing with memory, the role of computers and so on, circuits. And I found, to my amazement, that apart from the chapter on neurotransmitters, in every chapter, Crick muscled his way into it and turned out to play a fundamental role, not in doing the research, but in shaping the agenda. So our current interest in connectomes comes from an article that Crick wrote in, it was 1988 in Nature, because he was Crick, he could write a kind of absurd article. We must be doing this. This is, you know, throw out some crazy theory about dreams, for example, and get it published in Nature. John Maddox was then the editor. Go, oh, yes, Francis, that'd be marvelous. Let's have that on the, you know, as the major piece in Nature. This think piece from a guy who's very clever but doesn't know anything about dreams or whatever. He did write about dreams, and he writes there that, you know, he gets very cross. He says it's absurd that we know more about the macaque brain. There'd recently been a very high level connectome, but unbelievably complicated connectome. So it wasn't a neuronal, it wasn't based on nerves and big areas, but it's still very, very complicated. But we know more about the macaque brain than we do about the human brain. This is outrageous. It has to stop. And so he proposes, using his, brilliant mind and his consistent method, then let's try and understand how, this is before connectome was coined. The word was only coined in about 2003, simultaneously by two groups. We need to have a good map of the brain so we know where we are and where we're going. But what's striking is that, so for example, we have the connectome of C. elegans, of its 302 neurons. There is an incomplete brain connectome of the Drosophila larva, or rather of a Drosophila larva. So they have got a maggot, frozen it, sliced up its brain into bazillions of bits, and then spent thousands and thousands of person years. This project's been going on for about 15 years now, trying to work out the wiring diagram of this maggot's brain, this one maggot. And they already know by doing comparisons of certain areas that, not all maggots are the same. You might assume that with 10,000 neurons, they're going to be basically hardwired, but they're not robots. It's not always going to do the same thing. So that individual variability is there even in the simplest of organisms. So a bit like with the genome, we had the genome in 2002. Well, we didn't, we had a representative genome of a white man. We now know that there are, 800,000, more, I can't remember, there's 8 million bases more, some of them coding in African genomes than there are in European genomes. So there is no human genome, there's only genomes, and it's the variability that is really the interesting, and it explains some of our behavioral and other variability between individuals. So similarly, connectomes are only ever going to be kind of general, and this is the scaffolding and the interesting stuff. The detail is going to come from how individuals vary and how that variation contributes to what we perceive, differences in behavior and personality and so on. But to come back to my starting point, I think trying to start with thinking about doing a mammalian connectome. So where the mouse has got about 80 million neurons, so we've got about 80 billion, so it's a big factor of difference. I think even that is crazy, given the amount of time it's taken. And you should start and try and establish the principles. Also, it's going to be the workflow techniques. So it's actually a technical issue here. Just like with the genome, we didn't start with the human genome. We started with the animal genomes, not because, and bacterial genomes, not because they were interested, well, they are, but I mean, that isn't why the people who were doing it wanted to do it, but because that way they would develop the techniques, the workflow packages, the ways of analyzing the data that would then be useful for understanding the big genome, our own, with its 3 billion base pairs. Now, that is completely on a different order of magnitude, less than the problem we have in the brain, both in terms of establishing the map and making that map functional. Because the map, what's his name? Valentine, who was I forgot his first name. He was a contemporary of Hubel and Wiesels. He said, where is not how. So even if you could identify very, very precisely where something was happening in the brain, you wouldn't necessarily know how it was happening. And that for me is the interesting question, how it all works. So your maps need to be maps that enable you to manipulate the brain. Functional, maps of the human brain are very useful, in particular if you're going to have surgery. You want to know where not to cut to not cause handicap later on. But if you want to really know, you need to be able to manipulate those neurons to alter their activity. And certainly in Drosophila, that's one of the things you can do, because the techniques used for establishing, you can express particular genes in particular neurons. So you need genetic tools. It all gets very, very technological, but I think that's part of the challenge we've got in making good functional maps of model organisms that are model only in the sense that they stand in for a vast number of others, that we can then try and establish basic principles. But we're back to the lobster's stomach. we've got the connectome, we've got the whatever it is, the sequome, the RNA sequome, or however you say it, we know all the genes that are expressed in each of those neurons. We can identify them. No idea how it works. I don't think everybody should be working on a lobster stomach, although I'm sure Eve Marder would be pleased if there were a few more people looking at it. But that is the challenge. And the public, when you explain this to them, they go, okay, now I get it. And that's why my book is very open about saying what neuroscientists say in pubs and in conferences, that we don't know a thing. And my editor at one point, when he was looking at a draft, he said to me, so how does the brain work? I said, I don't know. Nobody knows. He said, well, you can't say that. Every brain book has got an explanation. It's got a theory. It works on, quantum mechanics or whatever, some complicated theory. I said, well, that's fine, but I don't have any such view. And I mean, one of the gratifying things about the book is that my peers, so neuroscientists, have overwhelmingly, if not unanimously, been very, very positive about it, which has been great because, and I think one of the reasons for that is that I don't have an axe to grind. I'm not a player in the brain. In fact, I don't actually study the brain because I study peripheral neurons. So I'm a neuroscientist, but I'm not studying the human brain. I'm not interested in consciousness, particularly, although there is a very good chapter on it. And I think that the fact that I haven't got an a priori starting point, I'm not trying to prove anything, except, my God, it's complicated, isn't it amazing, which is the real message, means that neuroscientists can find themselves in it. Because even if I might be slightly iffy about their field, in particular, say fMRI, it's, gets a bit of a, not necessarily from me, but it does get a bit of a kicking and eyebrow raising in terms of its degree of resolution. Compared to what I'm interested in, which is neurons. They can also agree with the critiques of other fields. So everybody can be happy with at least with most of the book, even if there may be a bit of the book, well, that was a bit unfair. But I don't want people to get the idea that I'm being rude, because I was incredibly aware that I was, getting a very high level view from doing a lot of reading and a lot of talking to people of areas that people had spent their whole careers working on. And these are very smart, very good people doing some brilliant research. So I wanted both to amplify that, to explain it to the general public, because this is primarily a book for the general public. It's not an academic book in that sense, but also to show that there are common absences in all these areas, and that is overwhelmingly that of the lack of a theory to try and explain what's going on. Now, maybe there won't be, I'm not talking about consciousness, I'm just talking about the kind of things we've been discussing here, computation, whatever. Maybe there won't be such a theory. I mean, I got very different views when I asked colleagues about this. Rubbish, it's just lots and lots of little bits that work together. Okay, well, that could be true. Oh no, there is this going to be, whatever, I don't know, Bayesian, whatever, people have got their own thing. There's none of that, because I deal in very simple things, single neurons. I don't have that axe to grind. So I think that is one of the reasons why it's people have been pleased with it.
Indeed, this is a great book, definitely. What I liked, I mean, one thing is that you describe past, present, and future of the neuroscience. So it's good even for people who are like really starting, or as you mentioned, also for general public. But also, that your book basically that shows the weakness of both scientists and humanity, where we could understand something, but either because of certain reasons that we couldn't pursue those ideas, or so for example, this monopoly of Aristotle ideas which stayed there for centuries. And these kind of these are, I think, very important lessons for the future of humanity. And the other interesting thing that I liked was the fact that I have 50 other books to read and 100 other scientists that I'm following. Literally, like, I have at least 60 scientist emails in my
Okay. Well, thank you for
that I
choosing
would
me.
like to contact in the future and probably have a conversation with them. So it's that great. That's really there. But about the solution, which is now the interesting part, right?
Well,
So I also do another series on origin of life where I talk to scientists, and I'm like, okay, because cell, when we think of, or simplest cell, which is again, it's one of the complex systems in the universe. But even if you look at that bacterial cell, and we can now synthesize all the components artificially. And this is what I asked them. What do you think? So, like, at what,
me about the origin
when
of
the time
life.
will come? Yeah.
Well, I mean, I assume that, I mean, again, this is even less my domain, but I do teach it to our first year students, but that just means that I only, I know it to a very low level. I know enough to impress the students, hoping they're not watching, without actually knowing a great deal about it. So I assume that, I mean, so one of the interesting things is that you talked about the cell. I mean, we can't start with the cells. Cells must have come later, which a lot of people get very cross about. But initially, you will have had replicating molecules. You need to think about your definition of life without getting hung up in it. And again, philosophers can help there in this kind of thinking. But you need to have the possibility of acquiring energy from the external environment. But that almost certainly means that you can't be anything. It's nothing to do with light. And one of the reasons you can't be light is if you're a replicating molecule like an RNA molecule that can not only replicate, but it can also act upon the world. So RNA acts as an enzyme as well. It's one of the great discoveries. I mean, RNA is their interest. DNA, everybody's obsessed with DNA. I mean, I've written a book about it. I mean, it is the most boring molecule in the world. It is a library. It just sits there. It is pretty much inert. It can't do anything on its own, whereas RNA is the exciting stuff. That's what's whizzing around and making everything happen. And so the theory which I suggest to my students, whilst emphasizing it's a theory and that we have no proof of this, is that initially you will have had probably maybe in tiny pores of deep sea rocks where there are proton gradients because of the upwelling of slightly acidic water from the Earth's crust. There you've got a proton gradient, you've got free energy you can get hold of as a molecule and use to power your very slow metabolic processes that will have taken place. And those replicating molecules are going to exchange products with the outside world, and through the process of very slow process of natural selection, then enable eventually the big break, which is from the RNA world or some other replicating molecule to the DNA protein cell world, which now dominates, but which is most definitely not what we began with. Well, we didn't start, you got to look back to an earlier system. How that happened, well, one of the striking things, of course, I mean, at Manchester, we have a fantastic course that any student in the university can do called Are We Alone, which is jointly run by our colleagues at Jodrell Bank, which is the university's radio telescope, who are really into SETI, the Search for Extraterrestrial Intelligence. So they teach about the physics and the politics of SETI and all the rest of it. I teach about the origin of life and the nature of life on Earth and quite how unlikely it is. And then we have also historians of science who talk about, well, why are we interested in aliens anyway? You know, anyway, what is it? Aliens in culture and so. Anyway, so what's striking is that, you know, so the big question is the Fermi paradox. If there are aliens, where are they all? And one of the answers to that is, well, they're all slime. So if you look at the history of life on Earth, what's very striking, one of the arguments why I will generally accept that the universe might be full of slime, is that life arose on Earth pretty much as soon as the conditions were right. After about whatever, roughly 500 million years, the Earth's cooled enough for there to be stable molecules. There's water on what seems to be important as a universal solvent. And life, it appears from the genetic data, pops up pretty much straight away. So life appears perhaps n = 1 so we have no variability here, appears to be quite straightforward. The next stage, going beyond that, is a lot more complicated in the development of the cell, and then above all the eukaryotic cell, which is just the most bizarre accident in the history of life, because one Tuesday afternoon, two kinds of cell bumped into each other and one ends up inside another, and that's the origin. To be very crude, that's the origin of the eukaryotic cell, and that just happened, as far as we know, once, in that all eukaryotic organisms can trace their origin back to that event, which will have probably been in a population of cells, not necessarily just one, but it certainly happened on a Tuesday afternoon around about 2 billion years ago. So it happened just once. And how many times have single-celled organisms been bumping into each other over the 3 billion years in which we've had a history of life? So that is just mind-bogglingly unlikely. And you got to remember, it wasn't eating. So I always get this wrong. The two forms of prokaryotes, it doesn't matter, I get it the wrong way around. They weren't eating each other. So eating, phagocytosis is a eukaryotic trait. So it wasn't something tried to eat it, something else that then survived inside it. It was just ended up with this weird kind of hybrid organism that turned into us. And we've slowly stolen the best genes from our mitochondria and stuck them in our genomes, and the mitochondria is left with this shriveled kind of, it's just turned into our slave, basically. It gives us energy. But without that accident, you wouldn't have any multicellular organisms. So for 2 billion years, the aliens whiz by in their flying saucer, and all they can see, if they could see such a thing, would be slime, just kind of moving around. Anyway, that's I've gone off on. So I think we're back to the set of accidents, right? So the point about that eukaryotic, and this is I emphasize to my students, the point about the eukaryotic cell, or even the cell itself, is that was not, or the eukaryotic cell definitely was not the product of natural selection. It enabled, it wasn't, there's no evidence that natural selection had been trying to get there repeatedly, and different experiments had taken place. It just happened, as far as we know, once, and it's an accident. Once it takes place, then natural selection can operate on that and produce the amazing range of organisms we see itself. But the event is, as far as we know, a chance event. So, random stochastic events, which are unpredictable and are so mind-bogglingly unlikely, given the history, as I say, how long life's been on Earth and how many cells have bumped into each other without just once forming a eukaryote. That suggests that the other answer to Fermi's paradox is that, well, the bug-eyed monsters aren't out there, because that just does not happen. Now, there may be, because the big argument is, well, the universe is very, very big, and I accept that. There are an awful lot of planets out there, and there are lots of galaxies. So somewhere there may well be other strange organisms, but they're not going to look like us, and they won't be flying in flying saucers, and we're never going to bump into them because they're too far away in time and space. That's my answer. That's my suspicion.
Yeah, actually, because in my case, I mean, of course, I was curious in evolution of life and biology, and then I realized that how important it is to understand origin of life. So I started a series on that.
Yeah.
And then I realized that how important it is to also understand about cosmos, like astronomy, right? So I started another series there, like to invite some astronomers and ask them different questions, how they are studying different exoplanets, etc.
Oh, that's
And
great.
there,
Well, I'll check out your stream, and we may give our students some of your videos to watch. That's great.
Sure. So the point there was because there are scientists that they are trying to study the weather on those exoplanets, and the fact that we can look at certain signatures, because these are all gases, right?
Yeah.
And if any planet would have a microbial life, those signatures would change in the cosmos.
Yeah, but we know, I think the real issue is we don't know anything about exogeology, right?
Yeah.
So that's the problem. This is the thing on Mars. So they find the methane spikes, and they get, well, methane can only come from bacteria. Therefore, there must be, I mean, almost certainly not. Then they're increasingly thinking that it's all to do with weird Martian geology, which would, I mean, you know, Mars is just not like the Earth. It doesn't have it doesn't have a molten core. It doesn't have a magnetic field because its core stopped moving and so on. The same thing with Venus. They all got very excited. I've forgotten what it was. They found the trace element in the upper reaches of the atmosphere. This was about 2 years ago, and the researchers were very careful about how they didn't say this is life. All they could say is, look, we found this weird stuff that on Earth is only produced by bacteria. So maybe in the upper, less hot regions of Venus's atmosphere, there are kind of, you know, bacteria floating about. Well, why not? Except it almost certainly now looks like, again, it's just weird geochemistry. So I think the Star Trek idea of orbiting from space and being able, or from a satellite, from a telescope, you know, millions of light years away, and being able to detect definitely signs of life, I think that is unlikely. Let's just put it that way, because of our ignorance about, you know, geochemistry on other planets. As we become more knowledgeable, that uncertainty will diminish, I guess. And it may well be that there are particular substances that are only produced by life, and then we might have a reason to send a probe or something. So I'm not against this kind of thing at all. I mean, it's fascinating. It's, along with the nature of consciousness, a big question.
Exactly. I mean, the thing is that so far we haven't been doing this, time course measuring of these biomarkers. It's just one spike that we get.
Well, that's what on Mars is. It was the repetition of it as it warmed up that made me think, aha, right. You know, as the seasons change, well, the bacteria now coming out of their kind of carbon dioxide ice freezing, or it's thawing, and they're, you know, well, yes, perhaps. But also perhaps that is changing the geology and enabling the gas to be released. I mean, I'd be very happy if we found something on Mars. It would be, and if it wasn't us, you know, either us in the sense that it's gone on one of our spaceships, or us in the sense that it's not a bit of landed from an asteroid, you know, meteorite that hit Earth and then goes into space and eventually lands on Mars. So that would be fascinating if it wasn't like us. And I mean, if it was like us, that would also be exciting, but for other reasons. We'd have to then work out who came first. Was it, are we the Martians? Was it, this is one of the hypotheses that, you know, this is a meteorite that hits Mars. Mars is the origin of life, and this rock tumbles through space, lands on Earth, plunges down into the early ocean, and finds this marvelous environment whereby the replicating molecules or whatever can then reproduce themselves. And that would be pretty cool as well.
Yeah, then the only problem would be then we'll have to figure out the geochemistry of the parent planet and to really understand the origin of life, which will complicate the issue even more, at least for us, right?
Yeah.
Anyway, so this was so we went a little bit off road.
Sorry.
So my question was so how are we looking at the solution of the brain? So are we looking at putting some Legos together, like, you know, we solve different networks and at the end we'll put them together and they'll work? Or because it can't be like sequential building, like this organoid approach that, I mean, but anyways, what are your views?
Well, I tend to think that's going to be the best solution. I mean, you know, if you want to understand a whole brain, you've got to start with something incredibly simple, because you're just not going to get anywhere. We're back to Crick's, you know, looking for the simplest solution, and ignoring that, saying, well, okay, we can't solve that. So this was his approach to studying the human brain, was let's look at attention. There must be some correlate of attention. That's what he decided was going to be the interesting thing. Now, attention itself is not a neutral word. It seems very simple. And he talked about spotlight, of, you know, focusing on a particular event or stimulus. And that's what you were going to find the neural correlates of. But leaving that aside, and he didn't succeed, of course, you know, but the approach was to say, you know, we will one day know what the nature of consciousness is. And this is a materialist article of faith, if you wish. I mean, I can't, you know, I can only say that there is no evidence that thought is anything else, anything other than matter. It could be, but I've got no reason to think so. But I can't explain it that way at the moment. So let's just put that to one side and focus on what we can do. And that, I think we're getting to the point that you just suggested, that is that there are going to be these bits, or perhaps a very small whole brain, either small bits of, say, the mouse brain, visual cortex, or whatever, that we can understand, and perhaps we can map using functional maps. So these are maps of cells that we can manipulate and predict what will happen if we alter their activity, and predict in a model, then do it in the animal, do it the other way around, try and find explanations, with all the time the lobster's stomach lurking in the background, because those two rhythms, we don't understand them. But I think that approach is going to be the best one. But the brain is not actually modular. It isn't Lego. It isn't my computer, because, as I've said, there is going to be these interconnections, which are absolutely fundamental, in fact, for how it works, how function emerges from this blob of squishy, incredibly densely packed neurons and glia. We all forget about the glia, but the glia are incredibly significant. They are doing stuff. They are modulating the activity of the neurons. They're not simply, I was taught, it's insulation, that's what the glia are. It's just, no, not at all. So we're back to the kind of limiting metaphors. And we now, experimental data has led us to realize that the glia are actually fundamental to the organization of brains of all kinds. Same structures have different names and different origins in different animals. But in the mammalian brain, glia are amazingly important. So I think that some kind of, despite not being entirely sold on localization of function, I think a modular approach, simply because that's the only way we can do it. We're not going to be able to explain everything. We've got to be satisfied with, it doesn't sell books. Your book has to explain the whole brain. You can't say, well, I've explained this tiny little bit of the brain. Nobody would be very impressed with that. But I think scientists would be. And if you could then accurately model that, that would be a tremendous step forward. And it's a bit frustrating. So one of the big arguments has been over the Big Brain EU initiative, which cost billions of euros and was, I submit, from a neuroscience point of view, a complete waste of money. And lots of us said this and got very cross. There was a huge petition when this money was spent, because it was actually a project, and I have colleagues at Manchester who've done very well out of this, thank you, a computing project. It was about building new computers. And the bit of the brain they were able to model, the idea was they're going to build a model of the rat brain, and the absurd promises that were made when it was launched about what they weren't going to do was, anyway, what they ended up doing was modeling a tiny fraction of what's called the barrel cortex of the rat's brain, which is to do with the whiskers, where the whiskers move, and we understand a lot about it. But it was just the neurons. They didn't model any glia. So it was actually not a brain at all. It was just bits of a brain. And it was part of a brain and only bits of the relevant cells. So they modeled the connections, and they found that it produced kind of rhythmic activity, which is pretty good, but that's not what, that's just part of what brains do. The activity of the neurons synchronizes and you get alpha waves and all the rest of it. So I think that you need to have it rooted in anatomical experimental data. So that has to be functional maps that you can manipulate, and and focus on discrete, doable problems, and don't try worrying, as Crick argued, we can worry about that in the future. Let's deal with this bit now. Try and incorporate simple approach to what is a very complicated eventual outcome. And we can put one, brick in the wall. You talk about Lego, you can talk about it that way, but the bricks and the Lego are interconnected. So in principle, that you've only got part of the explanation. You're not going to have you can't study this thing completely in isolation, because it is connected to the rest of the animal, and the rest of the brain first, and then the rest of the animal ultimately. So I think we've got to be satisfied with partial explanations, which is all science has anyway, because it's never the complete explanation. Newtonian physics is fantastic for putting satellites around planets. We use the same ideas that Newton established and were then later developed in the 18th century. But physics just keeps on going, and is we can understand at that level without fully understanding what matter is. So you talked about philosophers earlier on. I thought it was quite amusing that the Science magazine asked scientists what the biggest problems in science were. This was the beginning of the millennium, and the two issues that everybody overwhelmingly voted were the two things that philosophers have been worrying about for the last 2,000-3,000 years. So what is matter? Why is there stuff and not stuff? And secondly, what is consciousness? So the philosophers, they got a head start us on both those areas, so physics and neuroscience. And to be honest, at the moment, their explanations of both those things are probably just as good as those of the scientists. But in the end, we will get there and provide them with a definitive answer. It'll be interesting to see whether any of them got it vaguely right.
Yeah, I mean, the only thing that I can say is that these are the best times to live, basically, because we can have at least these sort of conversations. I mean, try to understand what is happening in the field, what are the major questions, what is the progress, and what is the reality. So to shape our theories or models, experiments in much better way, which wouldn't have been possible in the past, right?
Yeah, absolutely.
So that's the thing. And the other thing is that, of course, the scientists like you who spend so much time to write these kind of interesting books, where again, you get all the information that you need. And basically, I think that's just great for science.
Well, I think other parallel forms of communication like this are also extremely significant and very useful, both for scientists, for the general public, and for students to get up to speed with really interesting ideas. So it's been great talking to you.
Yes, so thank you so much, and thank you for accepting the invitation.
Well,
and
thank you for inviting me. It's been very interesting. I hope the audience hasn't gone to sleep, and you can fast forward. Yeah. It's too late now. We've got to the end.
Yeah.
Thank you.
Okay, thank you.